All right, Patrick, it's great. It's great to have you with us today. So thanks again. I'm gonna just open with just reading a very brief piece of your resume, so to ensure that we have everything right. So you can correct me right away if I'm wrong, and then we just get, we jump into the conversation, right?
Yep.
Okay, so you are currently principal scientist at Merck, and you work on the railway site in New Jersey. I've been there many times actually.
Nice.
It's an interesting campus. I know at some point it looked quite empty. There was some reorganization. I don't know if it's more crowded at the moment, or it's like it used to be in the old days or not. But
yeah, it's a little more crowded. And actually, we're consolidating the New Jersey site, so all the Kenilworth people are moving to Railway in the next couple of years. So it'll be bustling.
The first time I came there, it was just after the merger with Sharp. Yeah. I'm not as young as I look.
Pretty young.
Anyway, so you started. You're from Iowa, which is an interesting state, I would say. It's very interesting for to become an important chemist coming out of Iowa, and you actually studied there, right? You started at the community college in northern Iowa, and then you moved to Berkeley, prestigious group with Hartwig, where you did your PhD, and right after your PhD you joined Merck, and you've been there ever since. Is that correct?
Yep. Yeah. So I've been there about 7 years. Actually, yesterday was my 7-year anniversary.
Oh wow! Congratulations! 7 years is quite an achievement. 3 more years and it'll be 10.
Yeah.
All right. There's an unusual way we start our chat. It's quite obvious, Patrick, right? It's what we really like to try and understand is something that in science doesn't happen very often, right? You see the science, there's a bunch of name in a paper, you rarely see the faces unless you are in that specific field and you go to conferences and you meet the people, right? But it tends. What I always think is it's a shame to lose the connection between the person and the science. And if there's one really cool thing that we find out producing this podcast is that you can really see the person in the science. And that's what we really want to get to. So based on this, it's obvious to start from the history, right? So even the long history. So if we really look back at yourself and how you grew up, and how did you get interested in science in the first place? So how did you become a chemist, and why?
Yeah, it's a good question. I mean, something I think back on. How did I get here? So growing up, I mean, I just grew up in a typical, Iowan farm family, not farm family, typical Iowan family. Surrounded by farmland. And I worked my way through, high school and working kind of odd jobs at grocery stores or bussing tables, telemarketing. And I didn't really know what I wanted to do. I was an average high school student. So, I didn't want to spend a lot of money on college. So I went to the community college initially, which was near my parents' house. I had some interest in science. I didn't really know what I wanted to do. And it's probably a bad reason to choose a career, right? But I started watching like medical shows like House and Scrubs. I was like, oh, that seems pretty cool, and completely, oblivious to it. So far-fetched from reality. I was like, oh, I'll be pre-med. I'll go into med school and become a doctor. So I transferred to the University of Northern Iowa, I think in 2007, after 2 years of community college. And then I started taking, pre-med classes, biology, chemistry, organic chemistry. And then the organic chemistry class, it was pretty daunting because that's, like the med student killer, the pre-med killer. Like, oh, you take organic chemistry and you change your career. So, I was very intimidated. So I actually read the textbook ahead of time. It was like a 1,200-page book. I read it in advance, and something just sparked in me. Like, oh, this is so cool. The creative aspect, like the multi-step synthetic planning, just understanding how reactions work. It was really cool. And then, I started thinking about a career in chemistry, and I, over the course of a couple of years, I started doing undergraduate research with Martin Chin, who was, instrumental in, telling me to, do a PhD and giving me more exposure to research. And then from there, it kind of was natural progression into a PhD and then into pharma, process chemistry.
So it kind of happened by chance. Chemistry just happened to you in a way, on your way to becoming a doctor.
Yeah. And honestly, like when I was taking the chemistry class, I had no idea that there was even like jobs for chemists. I mean,
Of course.
you don't hear about chemists. You just hear about, medical doctors or other common professions. But I didn't really know what being a chemist actually entailed. There's actually
it is very
value
true.
in
It's
chemistry.
very true. It's and when they show scientists on TV, it's typically like mad scientists or someone like with superheroes and people trying to destroy the planet. But chemists are actually doing good things, most of them at least. So it's getting into from the community college. You did some work with Chin, and that's been a key mentor for you. And from there, getting into Berkeley for your PhD, that must not be an easy thing. So you must have had some sort of particular skills to get in there.
Yeah, well, actually, the funny thing is I did not get into Berkeley. So I applied to a bunch of grad schools, Berkeley, Harvard, MIT. I didn't get into any of those, but I did get into Illinois. So I actually started my PhD at Illinois. So John Hartwig was there at the time. So
it is true.
So I started grad school there. And then after a year or so, he announced that he was moving the group to Berkeley. So then I actually got into Berkeley kind of through the back door. And then I kind of joke with some of the professors that were on the admissions committee at the time, like, oh, you didn't let me into Berkeley the first time. But
well,
anyway, so
John Hartwig was a famous chemist even before Berkeley anyway, right? And, so it's still a prestigious group to get into.
Yep, definitely.
So what role did John Hartwig play in your development? That must be a crazy experience to work with such level of chemistry. Probably a big group with a lot of talented people. How important was that?
Yeah, so working in the Hartwig group was, a dramatic change from what I experienced, doing undergrad research, where it's basically just me and the professor. And now, the Hartwig group was, I think at the time, probably like 30 to 35 people, very smart people, very motivated, creative people. And it was just amazing to be around these people that, were doing, really cool science that was being published in, good journals and getting good recognition. And then obviously John has, developed chemistry that's used around the world and, really practical applications. And then, John Hartwig specifically, I think was a phenomenal advisor. He, takes training and teaching very seriously, especially, for young grad students, teaching them how to think critically, teaching them all the fundamentals. And there's a lot of things he does that goes above and beyond to, really train people to be scientists. So I'm extremely grateful for him. And then, when it comes time to apply for jobs, he's very supportive and, helps you, get in touch with people at companies and, helps with interview tips and presentation tips. So yeah, John Hartwig was an awesome advisor. I definitely recommend him to, any undergrads listening to go into grad school in his group. I'm
just curious, how is it in these, strong groups where you have a lot of people and a lot of talented people? Is it competitive or is it a supportive environment? How much the mentor and the leader is influencing this aspect?
Yeah, so I would say it was competitive in the sense that I think people were very hardworking, but they weren't necessarily competitive with each other. So I think it was still very collaborative. Like, there was, the older students would help out the younger students, solve problems or, figure out how to use the glove box or whatever. So I think it was competitive in the sense that everyone's, very driven and, wants to, work hard and publish a lot of papers and get a good job eventually. But I think the, interpersonal reactions were all very positive. I learned a lot from older graduate students and postdocs.
And during your PhD, you already were doing, high-level science. You have a bunch of publications, really interesting work there. And, I guess, the environment helps you out in raising your own level. Somehow you get there automatically because everyone around you, right? They do great things. Was there a moment where you understood that you could actually be a chemist full time? That could be your future in your career.
I get, the first year or so was pretty tough, to be honest with you, because there's just so much to learn and you see everyone around you, being very successful. And the project I was initially working on was, very complex. I didn't really understand kind of the overall goals and like how to actually see it through to the finish line. But fortunately I got switched over to another project. At the time trifluoromethylation was very in vogue. So I switched over to this project with a postdoc and, she really helped me a lot to figure out how to finish a project and how to publish. And from there, I kind of saw like, oh, okay, I think, being a chemist is reasonable. Like, starting a project, coming up with a solution, publishing it. But at the time, like in my first couple of years, everyone around me, or a lot of people around me, were really interested in going into academia. And even, my advisor, John Hartwig, was, pushing me to go into academia because, that's kind of the natural thing. But it never really resonated with me. And I was actually kind of distraught, like, oh, do I even want to finish a PhD just so I can, become a professor and do this, which was not that exciting. So, maybe in my second or third or fourth year, I don't know when, I started coming across, process chemistry papers that was, published in, whatever journal. And that was really interesting to me. It's like, oh, there's actual, practical applications of organic chemistry that can have impacts in the real world. And it's, it's not just publishing for the sake of publishing. So, once I started learning more about process chemistry, that's kind of motivated me to finish my PhD and then ultimately get a job in pharma.
So you decided that you wanted to do something real well before finishing your PhD somehow, or at least when you were getting there.
Yeah. So, early on in my PhD, I didn't fully see the value of getting a PhD if it was just to become a professor, which, obviously there's value in people becoming professors, but for me personally, it wasn't that exciting to me, so I wanted to do something practical. And that's, looking back to what got me interested in chemistry in the first place, was like the practical aspects, like the organic chemistry textbook from undergrad. It was really nice because throughout all the chapters, there'd be like specific examples. If you're learning about amines, for example, it's like, oh, here's some amines in drugs, and this is how, they interact, and this is why amines are important, yada, yada. So I think that's what really excited me. Like, oh, actually, organic chemistry has practical applications. So that was kind of the main theme throughout my careers, the practical applications of chemistry and, impacting, human health human health in my case.
It is really important, right? And it shouldn't be underestimated, because a lot of people really see, knowing that you can actually do something tangible, with your skills. Mm-hmm. It's not just a paper, isn't it? Or it's not just an NMR spectrum coming out of someone. It was, potentially, it's advancement of knowledge, but it's not tangible, is it? And to be honest, I have to confess, very often with my job, I'm in market development, and do I really produce anything concrete with my hands? It's not really. You do, and I know, at the end of the day, when you say, I've actually done something that could be useful for someone, is really important, and I can really see. Surprisingly, no, that's not the key for many people. So would you say that you're more driven by the role you can play in society and, the actual usefulness of what you do, rather than by pure scientific curiosity? Or how do you balance the two?
Yeah. So I would say my main interests are practicality and like actual impact. So I would say that's, 90%. But, doing things just purely out of, creativity or, some interesting mechanism or interesting finding. And that's definitely interesting to me. But what really drives me to, do my job and do these like side projects is actual practical applications. So that's what I think drew me to process chemistry and, continues to motivate me day to day.
From the same perspective, what else do you like to do in life besides chemistry?
So I've gotten into like building furniture recently, which again is a practical aspect of a hobby, I guess. So I build like tables, benches, stuff like that. And then I also like to travel. So my wife and I like to travel, around the world, do hiking and outdoors stuff. So yeah.
Did you have any time to do that during your PhD, or you work like 70 hours a week and it's totally crazy? You just want to die on the couch.
A little bit. We're working, 6 days a week, pretty much all the time. But we did take advantage of, the Bay Area, traveling around. California was, beautiful place to live. We did take a few small trips, but during the PhD, it was mostly work, to be honest with you. Right. But now, I think it's important to, take a break and take vacations every once in a while because, it's important.
I completely agree. And if you survive that, then you have a good chance to survive a lot of other things in life, right? And for the ones of us who have been through it, that's your experience. Your wife is also a scientist, isn't she?
Yep. Yeah. So she's a process chemist at BMS, which is down the street. Well, down the street from Merck.
Yeah, it's a few miles down the road. Yeah.
Yeah. Yeah.
Yeah.
Yeah. So she was a PhD student with Hartwig as well. So she was a year.
Oh, this is where you met.
Yep. Yep.
Okay. So it's okay. It's a laboratory romance. It's really interesting.
Yeah.
Well, you went then from a very prestigious research lab. You went into Merck, which probably is also not particularly easy. It must be a sought after type of job, right? And did you have Merck specific, or did you have like a few? Pharma was your thing, or did you want to go into process chemistry specifically for some reason? Maybe the practicality reason you were mentioning before.
Yeah. Yeah. So I was specifically interested in process chemistry. I didn't actually apply to any like medchem discovery chemistry jobs, which is probably risky.
Yeah,
but yeah, Merck was definitely at the top of my list. There's a few other companies that are obviously very good. But Merck was really at the top of my list. I had met many people from Merck kind of in my final year of grad school, and that was just, they were super smart and really nice and seemed like a great place to work. And so I was super fortunate to get an interview and land a job at Merck. And then once I got the job offer, I just canceled everything else, all my other interviews.
Merck is particularly exciting because of the attention they pay to technology, right? And being innovative and the sort of high level of science. I come from the biocatalysis field, and what they've been doing over the years in biocatalysis is just mind-blowing.
Yeah. Definitely.
And I know you actually have some experience with that. We'll get there later, but so you move from a place full of talent to another place full of talent, right? So is there any figure you met when you started at Merck, or colleagues or mentors that you met or developed, over the years in Merck? Is there anyone who has played a key role in your development when you actually started working at Merck?
Yeah, there was a lot. I mean, I think that Merck is definitely a intense place to work. It's, very fast paced and competitive. And as you mentioned, there's a huge focus on, innovation and new technologies. But that said, I think they also, what's really nice about it is they take a very serious view on like developing scientists and making sure people are successful. So when you start, it's very daunting, but there's a lot of people there to help you be successful, teach you how to become a process chemist, and, become an independent process chemist. So there's multiple people along the way that helped me. I don't want to list everyone because then if I leave someone off, they'll be sensitive. I would say the one person, had a huge role is my friend Kevin Maloney. So we've published multiple papers together, and this kind of happened kind of naturally. So when we first started, both of our offices were next to each other, and one day we just started talking, about chemistry and new reactions, and then this kind of snowballed into developing these side reactions, specifically making phenols, late-stage functionalization of sulfonamides, and a few other things. So yeah, Kevin, at the time, I think he had been there like 7 or 8 years. So he taught me a lot, not only about, how to run reactions, but, career development and just general advice. So yeah, awesome person, and he's become a really good friend of mine.
So there's a number of things I want to pick up on, and, I hope to remember them all. So moving from an academic research lab into process chemistry, there must be a complete change of, pace and word and objectives. On the one end, there's the sort of academic freedom of exploration, experimental space. On the other hand, you have a precise timeline, very specific cost targets to meet or efficiency target in your reaction to meet. It's probably fast-paced and very stressful. Maybe I'm wrong here, but there's a limit. There's a limit to the curiosity and creativity you can apply to it, because you actually need to get something done in a short time, right? And yet, particularly with your many papers with Kevin, you seem to have quite a lot of opportunities to develop new methods and produce innovation. That seems quite unbelievable for a process chemistry person. Is the normal American? Is everyone doing that?
I would say it's certainly supported, to have these side projects. It's just, honestly, looking back, I don't know how I did it, to be honest with you. It's, it's hard to find the time to actually do these things because, a lot of these things were completely unrelated to projects. So, during the day, I have a full-time job as a process chemist and then trying to squeeze in time, develop these, new methodologies and, write papers and do the ESI and all that. It takes a lot of work. Honestly, looking back, it's crazy that, what we accomplished. But I think what motivated us to do it was like, Kevin and I became friends, and it was actually just kind of, fun. Like we would get in the lab, for an hour at a time or so and just run reactions, just, chat and, come up with new things. And it was really fun. So it wasn't even like work, like during your PhD, you're just cranking through stuff. This was, fun because we were hanging out and running reactions. And then things I think can move a lot quicker in, a large pharma company because the resources are incredible.
Yeah, of course.
So if we want complex substrates, we can just order it from, a library, have it shipped to us so we don't have to do these, multi-step synthesis to make some substrate that may or may not work. And then, the analytical techniques are really nice. And, if we need help with purification, there's a group that can help with that. So things do move a lot quicker. But yeah, finding the time is not always trivial because, as you mentioned, we have real projects with real deliverables. There's real patients waiting at the other side. So.
Yeah, of course. And it's interesting the way you describe it because it seems to be motivated by passion for chemistry, a bit of a friendship component there, having someone like-minded, common interest, right? Something fun to do.
Yeah.
And I was kind of expecting that you had some practical motivations. I don't know, some problems in your process chemistry that you couldn't solve, and you were looking for some alternative methods, and that might not have been the trigger for it, but it doesn't seem to be there.
Yeah, so I would say there wasn't a specific project that we were trying to impact, but Kevin had been there for 7 or 8 years or whatever, and I'd been there for maybe about a year or so when we started doing this, and we had kind of noticed like a few things in general that were tricky. One of them being, making phenols, which on paper it looks like it's not really an issue, but in practice it's actually not trivial. And we saw, multiple projects where this was an issue. So then Kevin and I kind of came up with this idea to make phenols more general and, under milder conditions, better functional group tolerance and so on. And, we thought it'd be somewhat practical, but we didn't realize kind of the full potential. So actually, when we were developing, even before we published it, we started talking to people about it. And almost immediately, even before it was published, it was demonstrated on a couple kilo scale for a project where they needed to make a phenol and no other method was working. So that was kind of cool, like right off the bat. People were using it in process chemistry. And then, we told, the MedChem colleagues about it. We published it. And then, over the last few years, it's been used, hundreds and hundreds of times at Merck. So it's really become like a
beyond
go-to
Merck
method.
as well, as far as you know.
Yep. Yeah. So there's a couple academic, published examples of people using it to make substrates or in total synthesis. And then I have friends at other companies that say, they use it quite a bit, and their colleagues.
So let me get into some of the details here because this is interesting. So the way you make phenols is typically, is it like an aromatic nucleophilic substitution basically? So you start from the arene halide and use very strong basic conditions, and you do the nucleophilic substitution. Yeah, is the problem that when you start having complex multifunctional compounds, then you basically hardly have something which is tolerable of the reaction conditions? So you were looking for, was that the start of the project?
Yeah. It's mostly if you have, for example, an aryl halide, you can make the phenol via SNAR. Typically, if you're doing SNAR, typically you use like something like methoxide, and then you demethylate it afterwards, because hydroxide itself is not a very good nucleophile. You can use it in certain cases, and you can also do like CO coupling, with hydroxide. But the issue is, they're strongly basic conditions, high temperatures, and in real molecules, you're going to have functional groups that are not tolerant to that, especially things like esters or polymerizable stereocenters, where these just get destroyed. So we were really looking for something that could react under mild conditions, that would tolerate basically every single functional group you would ever have in a complex molecule. And that's where we came up with these hydroxide surrogate reagents, where they're oxygen-based nucleophiles, but they're quite acidic, so they react under mildly basic conditions. They're also great nucleophiles. So then they react, and then in situ they actually fragment once you make the initial CO bond to reveal the phenol. So it actually, we had a couple of examples, one with acetohydroxamic acid, and then another couple of papers with benzaldehyde oxime.
Yeah, so this, I've seen your paper with the oxime, so And it's interesting because then you use oximes for something else as well, because it seems like a very interesting creative thing, or lateral thinking maybe. And I want to go into that later. That's another point I need to remember for our conversation. This is already getting quite messy, to be honest. I need to do a better job. But that was really interesting. Is it true that people call it now the Molnupiravir reaction?
That's what people at Merck call it. I don't know how broadly used it is, but
well, we should publicize it. You should advertise it as, this is the Molnupiravir.
I agrre. Yeah,
definitely. Absolutely. So you have your own name reaction that makes you even more prestigious chemist. So you absolutely need to, to do that. It's really cool. So you mentioned it's been used over 800 times at Merck already, and it's already it's referenced, and there are several citations beyond Merck, and it's interesting. So well, do you think this might actually get some proper relevant space in the organic synthesis community?
I hope so. I think once people use it, they realize how powerful it is. So I think, as more people become aware of it and they actually use it, I think it should become more mainstream.
Have you used it yourself in any process chemistry work?
I have, yeah. It's been used in a few different process chemistry projects.
Okay, that's interesting. So let me step back a little bit, because some of our audiences might not be completely familiar about the workflow in pharma, right? So process chemistry is really that part of the work, and maybe jump in and correct me if I describe it wrong, where you take drug candidates coming from medicinal chemistry, drug discovery, and you actually need to make a process that can be put into a plant to make them commercially viable, right? And to scale them up, and having something which is robust. In the old days, medicinal chemists had to just use whatever methods to produce as many compounds as quickly as possible. So the chemistry isn't necessarily robust, it's just suitable to produce a big number of compounds and chemical diversity. So process chemists usually had to start from scratch pretty much. Is this still the case these days, or has the sort of culture or the pharma workflow changed a little bit or evolved in any way?
Yeah, so I would say it definitely is a case-by-case basis, but a lot of times like the goals of discovery chemistry are very different than the goals of process chemistry. So like the syntheses that med chemists are using could be more for SAR, or typically for SAR, where it's more divergent and complexity oriented, whereas in process chemistry you have a single target, so you just want the best way to make that one target. So in many cases the synthesis completely changes, and our goals are very different. In discovery chemistry, if your starting material costs $1,000 a gram, doesn't really matter, but in process chemistry you need something that costs $1 a gram, actually typically much less than that. So the goals are completely different, and a lot of times we start from scratch, but also we have a lot longer time to figure out a synthesis to a molecule. Where in medchem, you want to make multiple compounds a day, whereas in process chemistry, you want to come up with the ultimate synthesis over the course of probably a couple of years. So that allows us to invest in coming up with new reactions, coming up a lot of times with enzymes to catalyze reactions. Evolving enzymes can take a lot of time. So yeah, I would say in general, the synthesis does change quite a bit. Usually we cut the number of steps down significantly. We streamline things and make it robust for large-scale manufacturing. And then, new technologies such as biocatalysis and flow chemistry really come into play and really are powerful tools. Yeah,
of course. And I'm assuming you just rely on the specialists for that. So once you have something which is amenable for a biocatalytic transformation, so whatever other type of specialized catalyst, you probably have the specialist teams helping out in the project.
Yep. Yeah. So we have a really strong group of in the catalysis group and protein engineering that can identify the initial enzymes, do the evolution work, and really develop these enzymatic transformations. They're awesome team. A lot of them are trained synthetic chemists, or even process chemists that moved into biocatalysis later in their career. And there's also a team in flow chemistry that has expertise in flow chemistry and developing these processes where they can benefit from flow.
So what I was trying to go earlier was how culturally shocking was it to get from an academic lab when you kind of have your freedom to explore, and it's a tough job and you put a lot of hours in it, but to be thrown in a world where you have like a relatively short amount of time to come up with something commercially viable, or reinventing a synthetic road from scratch, with pressure and cost and knowing that there are patients waiting, right? So how tricky was that, and how long did it take you to feel comfortable in your job?
I don't know if I've reached that comfortable stage yet. No, sure. Yeah, I mean, it's a huge switch from academia, right? Because you have real deadlines, you have like real deliverables, and you're doing things that you're not used to doing. Coming up with like a large scale manufacturing process. So I would say, yeah, the transition can be pretty challenging, but there's people there to help you, like I mentioned. But there's a few projects that kind of stand out. About a year in, I was put on this project where it was this complex cyclic dinucleotide, which I had no experience with nucleoside chemistry, and this had stereogenic phosphorus, atoms. It was just extremely complex. And I was like, oh my god. And I'd only been there for about a year. It was just extremely complex. And this was a project where the med chemist did a phenomenal job making all these analogs, but the synthesis they were using, it was like 0.1% overall yield, and it was something like 12 steps, and a lot of multiple. It was a no-go.
It's a no-go.
Yeah, and the company wanted to move this compound into clinical trials, and we needed to make enough to go into a phase 1 study. So we're looking at this dozen step synthesis, 0.1% overall yield, and we need to make a couple hundred grams, and it was very daunting, but it was motivating because this is something where like doing chemistry can actually have a real impact. So quickly, me and a few other people came up with some better conditions, and we improved the yield maybe to like 15% or 20% yield over the, I think maybe 8 steps or so we got it down to. And then we were able to go down to our prep labs and do it GMP, a delivery suitable for human consumption. And yeah, it was super exciting. And then looking back, it's like, wow, that was awesome. It was very challenging, but we met the deadline, which I feel like we always do, and then we actually impacted human health. So
it's good. I think it's very healthy, right? Once you get there, just stopping and looking backward and say, oh, just made it, and this was really, really tricky, right? So I can actually do it. So these are typically the moment when you say, oh, I can actually make this. I'm actually quite good at it, right? It's always difficult to admit, but and that's what it really means, in my opinion, to get comfortable in your own role, right? And you're saying that you don't quite feel like it because there's always a new challenge, right?
Yeah. I think that's the other thing with industry versus academia. Like when you're in academia, you kind of choose your problem. You probably choose something that you're comfortable with, you're an expert in, or you kind of choose an arbitrary target. But in pharma, you don't, which I think is actually a plus because it pushes you outside of your comfort zone. You end up learning about all these areas where you had no maybe interest in, or definitely no experience with. For example, like nucleoside chemistry is very big, biocatalysis. I had no experience with any of these things, and now I've been involved with multiple projects, and I'm better for it.
Do you enjoy trying new things and getting into completely unexplored areas for you? Do you see this as learning, growing opportunity, or are you ever scared of those?
I think initially it's kind of daunting.
Yeah.
Like the first time I was using or doing a biocatalytic step, it was, I think from the outside it's like, oh, biocatalysis is so complex and scary. And
so is it alive?
Then you start, But then you start working on it, you're like, okay, this isn't that different than normal chemistry, and you can still optimize it in the same way. You can, obviously there's subtleties that make it much more complex in certain cases, but I think it's normal to be kind of maybe scared initially, but things are not as complicated as they may seem. And again, there's people there to help you, and there's experts that can help you figure out what's going on and come up with a solution.
Do you think the average synthetic organic chemist is a bit conservative, and they look at these sort of novel techniques with suspicion?
I think at Merck not, but maybe in the broader community. I think enzymes are becoming more and more mainstream, but I think there's still a huge barrier to doing that. Even other technologies like high throughput screening, which Merck's been involved with for, I don't know, 15 years or
time.
I don't even know when. But I think even that, like in grad school, we tried to implement that technology, and I think there's a huge hurdle to just doing it for the first time, like setting up a 96 well plate. of reactions just seems so overwhelming. But once you start doing it, you realize the power and the capabilities of that.
So the challenge is really setting up the work culture to be able to stimulate this sort of kind of daring approach. Right? You need to dare try, and you seeing the value and accepting it. And particularly with biocatalysis, when I think about it, it's been around for a long time. I mean, it's not a new thing by any means, right? But if you speak to 10 synthetic chemists, probably 9 have never touched an enzyme in their career, right? Maybe even more than 9. But it's a shame in many ways, but there must be a meaning. And obviously, as you say, Merck is certainly not like that. And I'm sure there are other organizations, or even academic teams that aren't like that. But do you think there will be an evolution in the community at some point?
I think so. I mean, I think people are starting to realize the power of enzymes, and I think more and more people will start using them. There's more academic groups starting to use enzymes and going to biocatalysis, directed evolution, and so on. So I think it's becoming more mainstream.
I'd like to jump on some others of your sort of lateral works, because your methods for synthesizing phenols is one. I was reading a couple of your works on the CH functionalization of pyridine. That it was also pretty awesome, and really, really, really interesting. And I noticed the commonality with the use of these oximes, sort of reagents, and I was wondering whether that is the common point between your work on the phenols and this one, or if it's a complete chance. It seemed to me like a potentially very interesting case of chemical creativity and lateral thinking.
I think there's a kind of common theme between a lot of the work I published at Merck is like breaking like nitrogen heteroatom bonds, a lot of times oxygen, but in other cases like sulfonamides cleaving the NS bond. So there's definitely commonalities there, but they're kind of serving different purposes. Sure. Like in the phenol case, like the nitrogen oxygen bond fragments reveal the phenol, and it also makes the oxygen more nucleophilic. And then in the pyridine case, it's acting as an oxidant, and then the sulfonamide case, like it's a way to cleave the NS bond. So there's definitely a lot of overlap there. At some point I was reading this. I said, well, this guy really, really likes these oximes, but it just makes a lot of sense, right? Because once you find the utility in a way, you're tempted to kind of look at it from different angles and say, well, maybe there's potential for doing something else with it. And I was wondering whether this was at some point, at any point, your logic, or if it just happened by chance, and maybe this is my passion for trying and understand the thinking process behind work, right? And how people get, because very often there's actually a practical need. So you need to solve a problem and you just try and then you find a solution. Or sometimes there's an actual systematic exploration of what you can do with certain things, right? Or with certain methods. And how do you find yourself thinking more? Are you more looking at it from the practical perspective, or is there any element of sort of scientific curiosity that push you to explore more with what you found out? I think for a lot of these projects it was identifying synthetic gaps or just like repeated problems that you would see, like functionalization of pyridines. A lot of times people make the pyridine N oxide and then functionalize two position that way, and that has its own challenges, strong oxidants, strong dehydrating agents, and kind of limited scope. So that kind of, and then actually in grad school I was also involved with some pyridine functionalization chemistry, but which was really cool, is like to fluorinate pyridines in the two position selectively, and then you can do various things with that. But that had its own limitations. It had strong oxidant, precious metal. So the motivation was come up with kind of a more practical way to functionalize pyridines, and also using something that's inexpensive and simple, and kind of a fairly unique mechanism, right, where you activate a pyridine towards nucleophilic addition, and then you have this intramolecular fragmentation to re-aromatize the pyridine.
And this is the process chemist thinking, right? You need something that is real and that can actually be applied. So just going to the chemistry a little bit, the approach is not particularly different from the more traditional going via the N oxide, because the idea is doing an electrophilic activation of the adjacent carbon, right? So that's the way it works. But you use this chloro, this is an aldoxine, isn't it?
Yeah.
So that binds to the nitrogens as you form the pyridinium salts, and then you basically have your carbo, which is more electrophilic, and you can use any nucleophile. So what kind of nucleophiles can you use there? What kind of functionalities you can add to the pyridine with this?
Yeah, so in the original Jack's paper, I don't know, 2017 maybe, it was primarily cyanide, and I think that works for a few reasons. One, it's a pretty good nucleophile. Yeah. But the other thing is I think it acidifies that CH bond adjacent to the nitrile. So when you initially add, you have that dihydropyridine adduct. So I think the cyanide helps acidify that to then be deprotonated and ultimately re-aromatize the ring. So there was a few other nucleophiles that worked okay, like proof of concept, malonate or methoxide. But what I was really interested in is amine nucleophiles, because 2-aminopyridines are extremely prevalent. So if you look, at FDA approved drugs in the last couple of years, I mean, a huge percentage of them contain 2-aminopyridines or similar functional groups. But the issue is, I tried hundreds of reaction conditions to install amines using that same activation concept, and it just didn't work. The amine would react at the oxime and just, kick out your pyridine starting material. Yeah. Or it would add to the 2-position carbon, but then instead of fragmenting how we wanted to, it would just open up into this, zinky aldehyde type thing where the pyridine ring would just snap open, and then it's dead end. So that's what led me and this guy Soo-Hong Kim, who is superstar intern with me a couple years ago, to develop what we call the Aminator, which kind of gets around these issues. And it was completely redesigning the reagent to basically make 2-aminopyridines from pyridines.
I understand. It's a really interesting work, and this is immediately evident, right? The utility is obvious, as you say, because of the ubiquity of sort of pyridine or aminopyridine type of groups, and so it's a no-brainer to understand why you went there. What was kind of surprising to me is the work you did on the sulfonamides, because the logic seems to be different. The way I've always looked at sulfonamides are more like a sort of terminal functional group. I never, maybe because I'm not a good enough chemist, but I never look at them as a sort of symptom, right? Functionality. Yeah. But with your work, it looks like it can actually be a pretty handy thing to functionalize. So you can change the substituent pretty much on the sulfur or on the nitrogen in a pretty flexible way there. So can you describe that work?
Yep. Yeah. So this came out again, like from another kind of random conversation Kevin Maloney and I were having one day in my office. So at the time, he was working on this compound that had a sulfonamide, a primary sulfonamide. And there was interest in making the same compound, but with a methyl sulfone there. So instead of NH2, it's methyl. So, instead of going back to the very beginning, going through this, 10-step synthesis or something to make the compound, we were wondering, is there any way to convert this compound that we have, 100 kilos of into this methyl sulfone? And on first glance, you're like, oh, that's impossible.
Yeah, you can't do it.
How could you, how could you do that? So, I don't know if we came up with a solution that day, but I think the next day, we came back with some ideas about, what if we can cleave the NS bond, how we eventually did it. And then you make a sulfonate, and then if you have a sulfonate, you can do whatever you want. You can try with methyl iodide, or you can do, various other functionalizations. So then this becomes a platform for converting sulfonamides into anything, other sulfur functional groups, or you can even remove the sulfonyl group entirely and do cross couplings or desulfonylative couplings. So this one came out of a fairly real need, and again, just Kevin and I chatting, and then coming up with a solution and going in the lab, trying a bunch of experiments, and fortunately coming up with a solution.
So once you go there, then it was let's explore what else we can do, right? So and then you develop it further. That makes a lot of sense. It's fascinating, Patrick, because there's such a healthy combination of scientific curiosity and practical needs, and you can see how the process chemist or the sort of academic chemist that kind of and probably Kevin as well, right? They kind of merge at some point. It's a fascinating process to me, and this is all beautiful work. Do you think this has the potential to become widely used? Because it seems to be of real practical utility. You just need to think about the chemistry in a different way, because people just don't think about using sulfonamide as a sort of intermediate, right?
Yeah. Yes. So I think, I mean, based on what's been published in the last couple of years since we published this, other people have kind of taken some inspiration from this and then come up with other methods for sulfonamide functionalization. And then internally, because I can look through our electronic notebook, or people just tell me, there's been multiple drug discovery programs where they have a sulfonamide, they have a lot of it, and then they can now use this chemistry to manipulate it and make other compounds. So it's been used quite a bit within Merck. I'm not sure about other companies. Actually, for our other work, so the initial work was primary sulfonamides, then we came up with a different kind of similar method for secondary sulfonamides. And this has actually been used as a way to deprotect sulfonyl protected nitrogen. So I actually saw like an academic presentation a few months ago where someone was actually using it to kind of remove an auxiliary that they put on their nitrogen. So yeah, different, it's just a deprotection reaction, but it's actually
it's definitely
pretty
useful.
simple way of doing it.
I still think that you could make for a great medicinal chemist as well. I know you don't want to go there, but
maybe not.
Maybe not. Yeah. Anyway, do you find more satisfaction from these sort of things? There's a nice dose of problem solving, or more from playing a role in something as big as developing the process for something that promised to end the pandemics, or at least play a role in it. I mean, I know I'm kind of going on a tangent, but I think we need to get there, right? So you work on developing the process for the Molnupiravir, the COVID-19 pill, as it's called. It went to the press. So yeah, you're kind of famous for that, aren't you?
Maybe a little bit. Yeah. I think both things are very satisfying. Like coming up with new methods that are used broadly is definitely exciting and motivating. But yeah, what really excites me is like working on developing a process that's used to supply patients with medicine around the world. Whether it's the COVID-19 molecule, Molnupiravir, which I can talk about, or other things I've worked on. I think it's super cool to as a process chemist, like you come up with the way that the molecule is going to be made, to supply millions of people around the world. It's just so cool that you go to a pharmacy, or someone goes to a pharmacy and gets a pill, and that pill was made using chemistry
I made
that you made. So
yeah, that must be something really. Yeah. It's particularly important, right? Obviously because of the time we're living. And for me, I'm part of Thermo Fisher Scientific. We sponsor the Talented 12 program. So you're a Talented 12 from last year. And Thermo Fisher Scientific is a partner of Merck in the manufacturing of this drug, and it's something you made. I really like this full circle, and I'm really proud of being chatting with you now today about this. But I'm sure there's some cool chemistry in there as well, and I know you actually work on two different syntheses. The most elegant of which is not really used commercially, at least not at the moment, right? But even before getting there, did you have like a tougher timeline and targets because of how urgent and important the problem was?
Oh yeah, absolutely. This was a project like no other, and it was also my first time leading a project. So, in I think May of 2020 or so, I got promoted to principal scientist, which is leading projects and leading groups of scientists. And then a few weeks later, I got assigned to be the chemistry lead for Molnupiravir. And this was, the fastest paced program in probably the history of, pharma. And yeah, it was awesome experience. And, it was my first time leading a project, and there was two efforts going on. So one of them was what became the manufacturing process. This is a 5-step synthesis from uridine. This has been used, I can go into more details. This has been used to make, I think, around 200,000 kilos of Molnupiravir so far, which is, many, many millions of treatment courses. And there was a second effort ongoing with a separate team, again, which I was fortunate enough to lead, on developing really the ultimate synthesis of Molnupiravir. And we came up with a 3-step route, starting from ribose and uracil, and it uses a couple biocatalytic steps and a really interesting glycosylation step, using some novel enzymes that never really been used before, and a way to recycle ATP with pyruvate oxidase and air.
So there's a cascade reaction there. Is it in vitro, or is it in a cell environment? Is it done in fermentation conditions?
So the enzymes are fermented and then isolated.
They're isolated. Okay.
Take those and then use them. Yeah.
So do you see this as being the future of the manufacturing? I know you made a lot of drug now and probably don't need to produce much more for in the short term. But I suppose the Molnupiravir might have like a broader indication as an antiviral going forward. Do you foresee Merck switching to the new roads to make it in the future?
Yes, I think it's possible at this point. As you mentioned, we're making it all with what we call the Gen 1 route. So based on how much we improved that route and all our manufacturing sites around the world, we're able to produce enough to meet demand. The second gen process is definitely shorter, and we've demonstrated on around 100 kilo scale. So at this point, we haven't moved that forward to commercialization because I think we can meet demand with Gen 1. But things may change if demand goes up or if other indications arise.
What is the reason why the biocatalysis route isn't the one you pick? Is it just you needed more time for development and it was just the time that was too strict? Was that the only reason?
Yeah, I think just the timing. Just to develop a process and demonstrate on 100 kilos is one aspect of it, but then you have to implement it at commercial sites. You have to do validation, and this takes a lot of time. And then of course there's regulatory components to it as well. So I think if that was basically it just takes more time, and by the time we kind of got it into a place where we could go into validation, the Gen 1 route was already validated and being used to make thousands of kilos a year.
You must be incredibly proud, though. Do you ever think about it? Did you wake up in the morning and say, "Oh, I played a role, a real role in the pandemic"?
I think back on it, it's incredible that I was involved with that, and the chemistry that I worked on in the lab with my own hands is now being used, to make enormous quantities of this medicine that's now being distributed around the world. So it's super cool. And then seeing it in the news, and the New York Times talks about this compound, the Wall Street Journal. So it's just super cool to have been a part of it.
Did any of these articles in the main press ever mention your name?
Not like the major.
Chemists are not popular enough, right? We need to do something about it. Maybe the podcast can help, but we need more. We need more. How many other drugs beyond this have you worked on that actually made it to the market? I'm just curious.
Yeah, so early on in my career, I worked, again, as just like an individual contributor on Zerbaxa. So this is ceftolozane sulfate is the active compound I worked on. So this actually was already on the market when we started working on it, but it's just the route was didn't meet our standards. So we kind of tweaked the route, developed a new process, and then refiled that. So this is an antibiotic that's been on the market for a while. And then another compound I worked on, relebactam, the MK code is MK7655. So this is a beta-lactamase inhibitor that's used for antibiotics. So I've been pretty fortunate to work on a couple things that I've seen on the market.
Well, I know a lot of chemists in pharma. They work whole careers and they never touch anything that actually goes to the market. So you've been good, probably.
I've been
more
lucky,
than lucky,
yeah.
But there's a luck component. And maybe working for Merck helps as well, I suppose. But congratulations. That's really fantastic. You must be proud.
Thanks.
You have to be proud. You should know. I would go on forever, but we probably have better things to do than speaking with me all day. It's you need to save the world. What do you think is the most important thing, in your job? Because it's incredibly complicated and there's a lot of components. So you certainly need a healthy dose of creativity, and you are demonstrating that you need discipline at the same time. So what do you think is the most important one?
It's a good question. Yeah, I think, from a scientist point of view, solving problems and, actually coming up with solutions to, real world problems. But I think what's also important is probably equally important is, working on teams and, really, working together in a team to like actually deliver this, the solution to the patients. I don't know if that.
No, that makes sense. Just, you're speaking about keeping in mind why you're doing it, right? But particularly when you're stuck in a problem and the solution isn't in sight, you must have a motivation somewhere, right? Do you ever feel like this is a problem I'll never be able to solve?
I think every problem's solvable, but the solution may not be as
obvious,
elegant as you initially planned. Yeah. I mean, there's always a way to like make something, but it may not be as beautiful as you put on paper.
And you need to make it commercially viable. So
yeah.
that's an important component to it. Now, I was reading on chemical engineering news that the Hartwig said of you that you make more interesting chemistry on a Friday afternoon than most chemists in the whole week. I can really see that after speaking to you. It was a courageous thing. Would you agree with him?
It might be a slight exaggeration. But, it was very, very nice of him to say that.
Yeah, from someone who produces, most beautiful chemistry on a Friday afternoon than any other chemist, in the whole week, and you've achieved already. You're still very young. You're just in your mid-30s, and you have 3 commercial drugs out there. You played an important role in solving the pandemic. I think it's a good time to come to my last question, which is always the same in my podcast. It's a good time to stop and look backwards and say, after all you've done, what would you recommend? What would be your suggestion to someone who is just starting in their career?
Yeah, it's a good question, and one I was trying to think of a good answer for. Send out the email. I think, looking back on what's been most impactful for me, not only getting through my PhD and getting a job, but now at Merck, I think having a strong network and talking with people, learning from others is so incredibly important. I think for students looking for a job or looking to do a postdoc, I think who and building connections with people is so extremely valuable. I owe a lot of credit to talking with people I met at conferences to actually getting me a job, interview at least at Merck, and then talking with people at Merck, just chatting with people, learning from others has been instrumental in coming up with these side projects. I mentioned with Kevin being a good example, and then talking with a lot of other people and learning from them and becoming a successful process chemist. So my advice is grow your network, learn from others, and don't be afraid to ask for help.
Thank you very much, Patrick. It's been a great pleasure, and it's a lot of fun speaking to you.
Thank you so much.
It's been great fun. Thanks a lot for being with us. Thank you.
Yep, thank you.